I will today discuss the mechanisms that contribute to the dissemination of SARS-CoV-2 into the body. And we will discuss how SARS-CoV-2 infection damages the airways. So as you know, the airways are the portal of entry for SARS-CoV-2. The airways are constituted by the nasal cavities, the pharynx, the trachea, the bronchi, and the bronchioles. And they connect to the lung alveoli where gaseous exchanges take place. It was shown early on that SARS-CoV-2 replicated efficiently in the epithelia that line the airways. And it can also replicate in the lung parenchyma, in the alveoli. This is in contrast, for instance, with the original SARS-CoV virus, which was identified in 2003, which preferentially replicates in the alveoli. So the capacity of SARS-CoV-2 to efficiently infect the airways may play an important role in its capacity to be transmitted efficiently. We wanted to analyze how SARS-CoV-2 replicates in its primary target cells in the airways. So to do so, we use a reconstructed bronchial epithelium model where bronchial tissue is put into culture, and the basal cells that are present in the epithelium will proliferate first in a submerged culture, and then the culture is airlifted, meaning that the epithelial cells are put into contact with the air, which triggers their differentiation. And you can see that then you obtain the different component of an airway epithelium. So here you have a photograph obtained by scanning electron microscopy showing a ciliated cell on the left and a goblet cell on the right. So you can see that ciliated cells, they carry about 200 cilia. The goblet cells here produce mucus, and you may distinguish the mucus globules that are ready to be secreted. So these two cell types cooperate in a process called mucociliary clearance, which contributes to clean the lungs and the airways. The coordinated beating of the motile cilia will move the mucus layer from the bronchiole to the bronchi, through the trachea. Ultimately, it will reach the pharynx where the mucus will be swallowed. So the inhaled particles that are stuck to the viscoelastic mucus will be cleared in this way, and this limits the accumulation of particles in the lungs, but also the propagation of inhaled pathogens such as bacteria and viruses. We next wanted to analyze the replication of SARS-CoV-2 in this reconstructed bronchial epithelium. This showed that there was a rapid replication of SARS-CoV-2 with already a 2-log increase in viral RNA at day 2 and a plateau until day 7. And this was measured in the apical compartment of our culture. To more precisely analyze the epithelial barrier function, we measured transepithelial electrical resistance by placing one electrode in the apical compartment, one electrode in the basal compartment. And you can see that tear measurements are fairly stable for an uninfected epithelium or mock infected, while there was a transient decrease at day 4 for the infected epithelium, but with already a recovery at day 7. So SARS-CoV-2 transiently impaired a barrier function, but with a fairly rapid recovery. Next, we analyzed the nature of infected cells by surface labeling with the SARS-CoV-2 spike antigen in green, most of the infected cells were labeled in red by beta-tubulin 4, which is a marker of ciliated cells. We saw only very few infected goblet cells or basal cells, consistent with most of the literature. We also noted that the infected cells in green here started losing their cilia partially or even totally, as shown here. When we look by scanning electron microscopy, it's very clear that the virus is destroying cilia. But you may see also that the continuity of the epithelium is maintained, consistent with the low amount of virus that we find in the basolateral compartment. When we look closer, we could detect the virus at the surface of ciliated cells. The viral particles were pseudo-colorized in green in these examples. So the structure of a cilium is made of an elongated axoneme, which contains a bundle of microtubules, and of a basal body, which is actually a centriole, which enables the growth of the microtubules. And you can see here that this infected cell, has already lost quite a few of its 200 cilia, but that the particles are not found on the cilia. Mostly they are found at the plasma membrane. We can say that it's not the replication of the virus that destroys the cilia, and that probably the mechanism for cilia loss is more indirect. We then look by transmission electron microscopy, and you can see that in a mock infected cell, you have many motile cilia that with the elongated axoneme and the basal body here that anchors the cilium into the cytoplasm. In an infected cells, you see fewer axonemes, and the basal bodies, they are not nicely arranged below the plasma membrane. They have various orientation, and often they are seen around vacuoles. Our current hypothesis is that these ciliated cells are undergoing a dedifferentiation, which leads to a loss of cilia, but maintains epithelial integrity. Now, we asked whether this had functional consequences. So we looked at the movement of polystyrene beads that were added on the top of the epithelia. And I will show you a movie showing that these 30 micrometers beads are quite moving efficiently in the mock infected epithelium. Now, if we look at the infected epithelium, you can see that the beads move much more slowly and that the tracks obtained during 1 minute are smaller. So we have an impairment of mucociliary clearance, which means that the mechanisms of cleaning of the airways is impaired. And we propose that this may facilitate the propagation of SARS-CoV-2 in the airways. I will thank the many people involved in this study. It was really a collaborative work. Thank you for your attention.